Welcome to the lecture series in food microbiology. In the last couple of lectures, we have been seeing what are the natural toxins or allergens which can be harmful to humans which consume them or to the animals which consume them, which are present naturally both in animals as well as plants. The next couple of lectures would also be based on natural toxins which are present in the plant kingdom. Curare. This is something which is very famous throughout history. Native people throughout the world have employed many natural plant materials as poisons to dip the arrows which are going to be used for hunting. So, one of the plant based material, a toxin which is being used by native people throughout the world is curare, and this has also been used in warfare during the prehistoric times. Even centuries before the arrival of Europeans, the indigenous human population of the Amazon basin in South America used extracts primarily of the climbing shrubs or the climbing vines, Strychnos toxifera and the Chondrodendron tomentosum. These two plants were used as sources of a potent arrow and dart poison known as curare. So, these extracts, the plant extracts, the arrows would be dipped or the darts would be filled, the blowing tips of the darts would be filled with these poison, and these were used to hunt animals. Crystalline D-tubocurarin, the primary active compound in curare, was isolated in 1935. The D-tubocurarin, it's when people started looking at its activity, it was shown to block the nicotinic acetylcholine receptors at the neuromuscular junctions to produce paralysis in the muscles. So, when the acetylcholine receptors at the neurotransmitter junctions are paralyzed, the neurotransmitter molecules no longer work. So, this in turn will lead to paralysis. This alkaloid first affects the muscles of the toes, the ears and the eyes, and then the effect will spread on to the muscles of the neck, arms and the legs. Finally, with high enough dose, the muscles of the lungs are also paralyzed. So, when the involuntary muscles of the lungs are paralyzed, the object would no longer be able to breathe. So, because of asphyxiation, the person would die. Recovery is possible, however, if respiration is maintained, which allows for the metabolism to detoxification. So, and then the excretion of the toxin. So, if you know that you have been poisoned by curare, then provided you can keep your muscles active, so that, because see, it is a slow paralysis of the neurotransmitter molecules leading starting to the finally the lung muscles are also paralyzed, leading to loss of respiration. So, without being able to breathe, the person will die. So, if you are going to keep the involuntary muscles of the lung active, then the toxin can be detoxified. Your metabolism has the capacity to detoxify curare, and then the toxin would be excreted. The curare alkaloids are not toxic following ingestion. So, in the GI tract, the efficacy is not much if you consume food containing curare, because their high degree of polarity inhibits their absorption for the GI tract. So, if you are poisoned with a dart or an arrow with curare, you will get paralyzed. But if you consume a food which has curare, then it will not be absorbed. So, apparently it is not toxic enough because absorption does not take part in the GI tract. Curare has been used as a diuretic and for madness, dropsy, bruises, edema, fever and kidney stones. The D-tubocurarin has been put to very effective use in Western medicine, however, starting in the 1940s as a muscle relaxant during surgical anesthesia. So, in the case of surgery, when muscles have to be relaxed, including the involuntary muscles, curare can be used as a muscle relaxant during the course of the surgery. Strychnine. This is also a very famous toxin which has been known from the prehistoric times. The first uses of strychnine were as a mixture with other natural products in a plant extract preparation, which again were used by the native people to the tips of the arrows and darts by native hunters since before recorded history. The principal source of strychnine is Strychnos nuxvomica. This is an evergreen tree which is native to Southeast Asia, especially India and Myanmar. The potent emetic activity of the extracts of this plant were very well known to the Western science by the time the species was named with Latin designation for vomiting nut by Linnaeus. The major toxic effects of strychnine progresses from flexed muscles to painful immobility, convulsions, and finally to complete muscle paralysis. The spasm of the chest muscles and diaphragm result in hypoxia and respiratory failure. So, this is an alkaloid, strychnine is an alkaloid and it is a highly selective neurotoxin. It is readily absorbed following ingestion. So, unlike curare, which is not toxic when absorbed or when ingested along with food, strychnine is readily absorbed following ingestion and it can also be inhaled, then also it can be a toxin, and then it can accumulate in the lipid storage sites in the body. So, what happens is that even if you are not affected by a lethal dose of strychnine in one shot, suppose you have been exposed like chronic exposure. So, exposure over a long period of time to small doses. So, what will happen is that this strychnine will get accumulated in the lipid storage sites. So, over a period of time, because of this storage, it can bind up or it can count up to the toxicity, because over a period of time this long time storage can lead to levels which are toxic. The chronic exposure to doses of strychnine that are not acutely toxic can eventually produce a toxic effect because of the buildup of the toxin in the lipid storage sites. Strychnine blocks the postsynaptic receptors of the inhibitory neurotransmitter glycine, and this neurotransmitter normally is active in the spinal cord and in the motor neurons. Glycine and strychnine bind to partially overlapping sites on the receptor. So, as you are aware, many neurotransmitter molecules have specific receptors for them in your body. So, among them, glycine has a specific receptor and strychnine also binds to the same receptor at the overlapping site. So, the strychnine sensitive glycine receptors and this receptor, glycine receptor, is actually a fast inhibitory is a molecule, or this receptor acts as a predominant carriers of fast reflexes of the motor neurons. So, what will happen is that the strychnine sensitive glycine receptors, which are the predominant carriers of the fast inhibitory transmission of the synapses in the vertebrate, spinal cord and in the brainstem. So, these receptors also belong to the ligand gated ion channel, which will similar to what we have as in the GABA receptor or in the acetylcholine receptor. Activation of this kind of receptor by glycine or other agonists will induce the opening of the anion selective channel of the receptor. So, as a result of the opening of this anion channel, you will have an inflow of chloride ions into the cytoplasm. So, from whichever cell you are talking about, here in the case the brain cell or the spinal cord cell, once the agonist binds to the receptor, it will open up the ligand gated channel, releasing from the lumen of the cell to the cytosol the chloride ions. This resulting hyperpolarization of the postsynaptic membrane will stabilize the resting potential of the cell and thus will inhibit the neuronal firing. Letharism. This is a very famous condition which is caused by eating certain legumes of the genus Lathyrus. The very famous legume among this is Lathyrus sativus. There are three types of letharism: neuroletharism, osteoletharism, angioletharism. Neuro indicating the neuronal transmissions, osteo indicating related to bones, and angio relating to cardiovascular activity. All of these conditions, the neuroletharism, the osteoletharism as well as angioletharism, are permanent conditions, but the activity, the symptoms as well as the affected tissues differ in each condition. Neuroletharism. This is a type of letharism which is associated with the consumption of legumes in the genus Lathyrus. So, you have multiple species of legumes in the genus Lathyrus. So each species is capable of causing one type of letharism. So here the neuroletharism is caused by the consumption of legumes in the genus Lathyrus that contains the toxin what is called as ODAP. This ODAP ingestion actually results in motor neuron death, and the result, when the motor neurons die, then there is no longer any signal transmission either in the muscle cells, or it results in paralysis and muscular atrophy in the lower limbs. So this is the Lathyrus species which causes that. Osteoletharism, it is a different type of letharism which affects the connective tissues. So it doesn't affect the motor neurons here like the neuroletharism, but here it affects the connective tissues, and this is caused by the consumption or the ingestion of seeds of Lathyrus odoratus, otherwise called as sweet peas, and this is often also called as odoratism. It is caused by a different toxin which is called as beta aminopropionitrile, which affects the linking of the collagen, a protein in the connective tissue. So connective tissue has huge amount of collagen. It is this collagen which is affected by the beta aminopropionitrile, which in turn is present as a toxin in Lathyrus odoratus. In angioletharism, the toxin involved again is beta aminopropionitrile. Just like in the case of osteoletharism, the toxin is the same, but the activity is in the blood vessel, not in the bone as in the previous case. Eating the chickling pea with grain having high concentrations of sulfur based amino acids reduces the risk of letharism if the grain is available. Some states in India have banned the sale of Lathyrus legumes in order to prevent the consumption of them, which in turn lessens the possibility of letharism in general population. See, most of these Lathyrus species are common legumes which are used as food. So the prevalence, especially in some areas, these are consumed majorly by the population. So in such pockets, you will have more predominance of this letharism happening. So in many states, it has been banned, where the sale of the seeds have been banned, so as to prevent the consumption, which in turn, so if there is no consumption, then there will be no onset of the letharism in general population. Atropine. Atropine is a neuroactive natural product, and its use has been known, both illicit use and illicit use, from the prehistoric times. So it has a long history, intriguing history of both uses. Atropine occurs with several other tropane alkaloids, prominently in plants of the Solanaceae family, primarily the Mandragora officinarum, and this plant was earlier called as Atropa mandragora, and another plant which is called as Atropa belladonna, the more predominant or the more famous counterpart, which is from which atropine has been extracted. This is the picture of the plant since prehistoric times. The term belladonna, meaning beautiful lady in Italian, it is usually thought to be derived from the practice of fashionable women dating back to Cleopatra in Egypt in the 1st century of BCE to use plant extracts to dilate their pupils for cosmetic purposes. So atropine earlier, the use of atropine to dilate the pupils was well known from the time of Cleopatra, and this was used as a cosmetic. The use of these plants is thought to date back to prehistoric times, and the written records indicate that the plants were put to rather sophisticated uses in ancient Greece and in ancient Egypt. For example, the book of Genesis in the Bible, 1400 BCE, it describes the use of mandrake to promote conception. Artifacts from ancient Egypt dating from about 1370 BCE suggest the use of these mandrake plants as an aphrodisiac and analgesic agent. The early Greek scholar Theophrastus, he stated that mandrake is useful for wounds, skin infection, gout and sleeplessness. So dating from the prehistoric times, from the cosmetic effect by Cleopatra and subsequent population, then from the medicinal use, it has been, the use of atropine has been very well recorded in the history. Around 35 BCE, Mark Antony's army is said to have been poisoned by Atropa belladonna, and also the Cleopatra used atropine containing plant extracts to dilate her pupils. Studies of the isolation of the active principle and the mode of action of belladonna and mandrake proceeded relatively quickly following the establishment of these plants as a bona fide medicinal usefulness. So, since there was recorded evidence of the medicinal properties of the extracts, the studies on these plants, how to separate the active component from the given plant extract, what is the active component, and then what is its, how does it scientifically act. These studies actually have been very well established. Using the meridiatic activity as a guide, the primary active principle, atropine, apparently was first isolated in a pure crystalline form from dried belladonna root in 1831. This substance was first synthesized by Nobel Prize winning German chemist Richard Willstatter in 1901. So, the active principle, the atropine, was crystallized in 1831, but the artificial synthesis happened in 1901 by the Nobel Award Prize winning German chemist. The natural product in atropine is a racemic mixture of D and L hyoscyamine, and most of its physiological effects are due to the L hyoscyamine. The toxic symptoms of atropine include dry mouth, drowsiness, dizziness, constipation and nausea. The other side effects are pupil dilation, blurred vision, fever. This fever is caused because the body is not able to perspire. Normally, if you sweat, the body temperature will fall. So, at high temperatures, you tend to perspire more. But when you consume atropine, you lose the ability to perspire, as a result of which temperature will build up. Inability to urinate, heart arrhythmia, and excessive dry mouth and eyes. The higher doses can produce sensations of burning throat, delirium, restlessness, and mania, including hallucinations. Death will finally happen with constriction of the airways and suffocation. So, finally, with slow build up of the effects, the airways will become constricted, you will not be able to breathe, and death will happen. The effects result from the atropine's ability to lower the rest and digest activity of the muscles and glands regulated by the parasympathetic nervous system. Atropine is a potent competitive antagonist of the human muscarinic acetylcholine receptors. Of the many recognized activities exhibited by atropine, including narcotic, diuretic, sedative, antispasmodic and meridiatic, the most important uses currently in the treatment of eye diseases and as an antidote to anticholinesterase nerve agents that have been used in recent times in chemical warfare and terrorism. So, if you go for an eye checkup, normally the doctor will prescribe some eye drops so as to dilate. This happens normally for any eye checkup. So, for that what we have is a extract similar to atropine because of its ability to dilate the pupils. For treatment of many eye diseases, we used similar compounds. Phytoalexins. Phytoalexins are plant metabolites that are produced by the plant in response to environmental stresses. So, when the plant environment relation is a dynamic relation which keeps on changing depending upon the environmental conditions at that particular point of time. So, even though the plant, it is a living thing, so depending upon the temperature, the stress, the soil condition, the climatic conditions, all over, the plants will tend to adapt and grow to survive in that particular condition. So, in times of environmental stresses, to withstand that particular stress, the plant will produce some secondary metabolites. These metabolites are commonly called as the phytoalexins. Invading organisms such as bacteria, viruses, fungi and nematodes can induce the production of phytoalexins in plants, with the result that the growth of the invading organism is inhibited. So, similarly, if the plant is attacked by pathogens, in response to that pathogen attack also or pathogen invasion also, phytoalexins can be secreted, which in turn the action of this phytoalexin to stop the growth or proliferation of the invading pathogen within the plant system. So, this can be induced in the plant either as a response to environmental stress or is a response to pathogen attack. This phenomenon is a general response to stress. However, exposure to cold, ultraviolet light, physical damage, and certain conditions or certain chemical compounds such as metal salts, polyamines, and certain pesticides can also elicit the production of the same phytoalexin in a given plant. So, each plant is inbuilt to secrete the phytoalexin as a response to a particular stress or to an invading pathogen. But the same phytoalexin, the same plant can be induced to secrete or produce the same phytoalexin when it is exposed to cold or UV light or physical damage or exposed to chemical compounds. So, we can use these to induce the production of the same phytoalexin. Classic example of the production of these so-called stress metabolites or natural pesticides. See, if the phytoalexin is secreted against a pathogen, then it means that it is acting as a pesticide against a particular pathogen. So, this is a natural pesticide. For example, very well known case in the potatoes, which are inoculated with the blight fungus Phytophthora infestans. So, potato blight, we must have heard of the Irish potato famine, where you had fields of potato in Ireland which were infested by Phytophthora infestans. They had a huge loss. So, those plants, normally when the infestation is less, the plant is normally triggered to produce huge amounts of this phytoalexin, so that the spread of the fungus no longer works. So, when administered on the surface of a potato slice, certain strains of Phytophthora infestans will initially grow rapidly, and then their growth rates will gradually decrease, because the plant will start secreting the phytoalexin against it. If an extract of the infected potato slice is placed in contact with a pure culture of the same fungus, then the fungus will not grow. So, you have a potato slice where you have inoculated with Phytophthora infestans. So, initially there will be rapid proliferation of the fungus. So, then the potato will start producing the pesticide, because of which the spread of the fungus and the growth rate will become slow. So, this potato slice is now infested, it also has the phytoalexin. So, when you expose this infected potato to a pure culture of Phytophthora, then the fungus will no longer grow there. This response to a variety of fungi has been observed in other plants like peas, green beans, broad beans, soybeans, carrots, sugar beets, cabbage and broccoli. So, this is a broad response of phytoalexin, production of phytoalexin against a particular pathogen, which in turn can also stop when in contact with the pure culture. It can also inhibit the pathogen in the pure culture. The plant responses in many of these cases are triggered by certain polysaccharide components of the fungal cell walls. So, fungal cells have chitin as well as other polysaccharides, and the plant has cellulosic cell wall. So, certain polysaccharide components of the fungal cell wall will trigger the response in the plant to produce the phytoalexins, and the quantity of phytoalexins which are produced by the plant can be very remarkable, in the sense that it is quite high. The chemical structures of these phytoalexins are generally modifications of the structures of the metabolites produced in unstressed plants. So, as we said, it is in response to a stress. This stress can be environmental, this stress can be because of invading pathogen, that you have the phytoalexins produced in. So, what happens in a non-stressed plant? Non-stressed plant also has certain chemical components. It is those chemical components which have structural modifications happening to produce the specific phytoalexins, which in turn will act as a protectant against the invasion or against the particular stress content. Because of their chemical diversity, abundance, the pesticide-like activities and the presence in the feed and food chains, the effect of phytoalexins have been very well studied. So, they are produced in large quantities, extremely diverse, because they are just modifications of the normal components which are present in the plant, highly effective pesticides, and since they are produced by plants, any plant, see, when a plant is growing up, if it is going to produce a phytoalexin in response to a particular stress, that phytoalexin is going to be present in the plant or the plant product when you consume it. So, it is present in the food chain or in the apparent food web. So, you need to know whether the consumption of this phytoalexin is going to be toxic or allergic. What sort of an effect does it have when humans or the animals consume this particular product? Consumption of moldy sweet potatoes in the field is known to produce severe respiratory distress, pulmonary edema, congestion and death in cattle. Two of the compounds, ipomyamaron and ipomyamarinol, cause liver degeneration in experimental animals with lethal dose 50 is equal to 230 milligram per kilogram. The lung edema factors from the infected tubers include 4-ipomyanol, 1-ipomyanol, ipomyanine and 1,4-ipomyanol, with ipomyanine being the most toxic, where the LD dose is 26 milligram per kilogram. The toxic terpenes from the damaged sweet potato can occurring only slightly damaged tubers used for food. The presence of these substances is associated with a darkened transverse ring below the skin of the tuber, and the liver toxin which is present here is the ipomyamaron. This was shown to be present in commercially available sweet potatoes at levels ranging from 0.1 to 7.8 milligram per gram of sweet potato, which means that the dosage will build up high. So, it is present in the food chain as well as the food web. So, in this particular lecture, we saw some of the natural plant toxicants which are present commonly, which have been used from prehistoric times, either as a poison, for example, strychnine and curarine, or an atropine which has cosmetic purpose as well as a medicinal purpose, to those which can act as a natural pesticide. Question in this particular episode would be, what would be the precautions you take if you see that the food product, for example, potato, sweet potato, rice, has been infested with a particular fungus? What would be the sort of storage as well as the precaution during the processing which you will take during this particular episode? Thank you.